Okay, what is this? This is July 22nd.
Yeah.
All right, so the first thing that we're going to do today before we start talking about the lab, because this lab is a pretty short one, it's got a lot of steps to the protocol, but it's the most cookbook lab that we've got, where it's like, just follow the steps carefully, and if you follow the steps, you'll get results. It's got centrifugation, it's got filtration. It's a pretty mellow one. DNA extraction lab, that is. But because we've got a tiny bit of extra time, I want to give you an idea of what you can expect on that lab midterm, okay? So I'm gonna write some bullet points on the board, but you can ask me if you'd like more details about what I mean by any of this. Okay, let's see if I'm in frame for the video, starting about here and going to about here is all in frame. Perfect. Okay, so I'm basically gonna run through some bullet points. Okay, ready? Ask me questions if you got them as we go. Solid? This is for the midterm that's coming up tomorrow. Okay. Convert between units. What do I mean? If I give you something in grams, you better be able to convert it into milligrams, micrograms, etc. Or vice versa, right? Milli, micro, that's all the stuff we worry about, okay? And nano, but just a little bit. Okay, molar masses. All right, given periodic table, e.g., calculate molar mass of CaCl2. All right, if you know how much each one weighs, how do you calculate the molecule, the whole mass of the molecule? Boom, boom, boom. Next, preparing molar solutions. Preparing specific volumes of molar solutions. Cool. Make me 400 milliliters of a 3 molar solution of calcium chloride. Okay. Dilutions. Dilutions, dilutions, dilutions. All right. Basic terminology. Oh my God, that's horrible chalk scraping. Okay, basic terminology like 1/10 equals 1 unit undiluted plus 9 units diluent. All right, so you need to know, like, if you took 10 milliliters of a solution and added it to 90 milliliters of water, how much did you just dilute that? And it's like, you did a 1/10 dilution. All right? Cool? You also need to be able to figure out, like, okay, hey, you took 5 milliliters of a solution and you added 15 milliliters of water. What dilution is that? 5/20 would get you partial credit, but it's a 1/4 dilution. All right, just simplify it. Question? Nope, that's good? Okay, cool. Boom. Next, estimating size of object in a microscope. You got to be able to do that. All right? I'll give you a field of view at some magnification, and I'll show you a picture on your quiz and say, like, how big's the thing in this field of view at this magnification, right? It's all stuff that we did, and we had a whole lab about it, you know what I mean? Cool. Tonicity / osmosis. That's going to be on there. Defining hypertonic, hypotonic, isotonic solutions and predicting what will happen to cells if put into different tonicity solution, right? What happens if you put an animal cell into a hypotonic solution, for example? Cool? Pretty straightforward. Dilution question. Dilution question. All right. Including serial dilutions. All right. Don't forget about serial dilutions. So if you do like a 1/2, and then a 1/5, and then a 1/20th, all right, that's a 1/200 total dilution. Okay? Nothing too crazy there. You need to know how to do, like, if I tell you, here's the starting solution, I would like you to make a solution, an end solution, with this concentration in it. How do you do the dilution? All right? So C1V1 equals C2V2, C sub I times D equals C sub F. All pretty fair game questions. But if there's one topic that's covered the most on the lab midterm, it is dilutions, because over the years of teaching this course, it's what people have the most trouble with. And you can't be weak on, hey, how's it going? You can't be weak on dilutions moving forward, because people will get frustrated with you asking questions about, like, could you explain dilutions to me? It's like, you're already working in the lab. This is something you should have a handle on. Moving forward, standard curves. All right, I give you a standard curve. I give you an absorbance value. You tell me the concentration. No tricks, right? Then maybe I trick you. No, I don't. I try to trick you in the final. In the lab final, I try to trick you by saying, you do a 1/20th dilution, and then you measure this absorbance. What was the concentration before the dilution? So it makes you do dilution and standard curve. This one's going to be the most basic, bare bones standard curve questions. You got to know what they are. Cool? All right. Boom, 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 boom. That's like, okay. Stock solutions. Don't forget what stock solutions are. E.g. 100x stock. Okay, remember this terminology. If you start with a 20x stock and you have to make me 5 milliliters of a 2x stock, how do you do it? All right, it's just C1V1 equals C2V2 stuff. This is just relative concentrations. Cool. I'm going to write specifically the entire nutrition facts problem from your carb assay protocol. So at the end of the carbohydrate assay protocol, there's a practice problem where there's a nutrition facts sheet, and you have to fill it out. I have to figure out how much do I dilute this to make it fall on a standard curve. All right. I rewrote it for the lab midterm because I think that brings a lot of things together, you know, and I like that question. I like it, and I'm not getting rid of it no matter what you say to me. All right. There's definitely something you could say that would make me remove it, but, you know, it would have to be just so mean that I quit this job. Which I know, you know, they're all capable of. You, people who are younger than me, you know my weaknesses. All right, moving on. We've got 2 questions that have to do with today's lab, DNA extraction lab, okay? It's going to be A260, A280, A2 260 to A280 ratios. The colon means ratios. All right, cool. Don't worry, we're going to cover all that in today's lab, but just make sure that you pay attention to that aspect. There's a question here about solubility and R sub F values in chromatography. All right, so remember, like, more soluble molecules move further if the solvent's nonpolar. Remember, we used a nonpolar solvent when we did our pigment extract, and then carotene went the same distance as the solvent because it's nonpolar molecule, small nonpolar molecule. All right, so what determines how far molecules move in chromatography? Well, it's a combination of solubility and size. All right, more soluble, moves farther. Smaller, moves farther. Questions? And the last thing that is on your, oh my gosh, it's so embarrassing. I just printed out 23 of these and the numbering's off. So there's 16 questions, but yeah. The last thing has to do with enzymes. All right. The last question has to do with enzymes. It's worth 2 out of your 50 points, and it's a trickier one where you have to look through your protocol to find the information. The information to answer all of these questions is in your protocols. So I put one question on here that's essentially testing your ability to look through a protocol for relevant information. I know, it's mind-blowing. 2 out of 50 points. It's 4% of your final grade. Question?
I know you said for the lecture midterm, like if we score a lower grade, we can make up points. Does the same go for the lab?
The same does not go for the lab midterm. Instead, I have it be worth, the initial one be worth fewer points, and, you know, be open notebook. Yeah. It only goes for the lecture midterm. I have made like one exception to that in the last couple of years for a student who had a whole like external issue, you know? So if an emergency happens, you know, you can talk to me and we can make exceptions and adjustments. But barring some unforeseen circumstance, there's no extra credit for that. Yeah. Any questions about any of these points? But thank you for asking. It never hurts to ask. Yeah. That's all that's on the lab midterm. It's a lot, but it's everything we've talked about so far, you know? Cool. Let's go ahead then and get into today's actual lab, all right? Which will be included on your lab midterm, which is tomorrow during our regular lab time. Oh, does anybody want to like take a picture of this before I put the screen down? No, everybody's good? You want one? Yeah, go for it. I'm not sure I'll have time to get the recording posted tonight, so if you know anybody who couldn't make it to lab today, then please share this information with them. Yeah.
You said there's something that's based off of the last question on the carb assay quiz. Did you have those graded?
The carbohydrate assay quizzes? No, those aren't graded yet. No. But it's what we did in the lab. So the question is on what we did in the lab, where you had to write it all out and do it for the lab to work. Taking the nutrition facts, calculating what the final concentration will be. That's what I mean. Not from the online assignment, from your protocol. At the end of the protocol, there's a picture of a nutrition fact sheet, and it asks you some questions about what's the concentration in micrograms per milliliter, and then how much would I have to dilute it. That's what I'm referencing. Yeah. And sorry, I don't have those lab assignments graded just yet. Also, I caught up with everything this weekend, but then assignments come in on Sunday at midnight, and then the week starts. You know what I mean? Maybe I should be doing due dates as Friday at midnight. It's like a grade during the weekend. But then that kind of messes with, I think that all of you probably need more time to take a break. Anyways, I have this debate with myself every semester. I was like, what's the better due date? Some people think it's Friday. Some people think it's Sunday. Most students seem to think it's Sunday. And I tend to agree. Question. You get the full lab period. So here's the lab midterm. Here's what it looks like. It is 4 pages, single-sided. So the one that you'll be getting is like 2 sheets, double-sided. All right, it looks like this. And you get 2 hours, 50 minutes to do it. Yeah. Oh, wait, sorry. 16. Yep. This is to give you all the time you could need to go through the protocols and look things up, you know. You won't fail this lab midterm because the information's not available to you. It's a mental game with this lab midterm of feeling overwhelmed, you know. And if that hits you, then it's like, oh, damn, I don't get this, and now I can't do the other things, you know? So be prepared to take breaks if you need them. Go walk up and down the stairs, breathe, have a lab notebook. There's always one every summer who just says, I don't need it. I'm not gonna do a lab notebook, I can just do it. It's like, sure, maybe you can, but why? Why not have the reference materials? Don't be the one. All right? Okay. Any other questions about the lab midterm? Sick. Today's lab. This is like, this is how a lot of Bio 105 labs, if you've been in a bio lab before, if you've done a lot of lab work in a larger class like this is what your lab protocols tend to look like. You walk in, you get this document, your instructor says, all right, now start at step 1 and go through step 16, and then you're done for the day. Have a good day. You know, and then I sit up here and I look at my phone. This is a cookbook day, where all you have to do is follow the steps well. And that is an important skill. All right, plasmid DNA purification and quantitation. So today what we're going to be doing is taking E. coli and we're going to be isolating its DNA and purifying its DNA. If you want to say sequence a cell's genome,